Hello, everyone, and welcome to Chapter 2 of Human Physiology. This chapter is titled The Chemical Level of Organization. Chapter 1 saw us focusing a lot on the fundamentals of physiology, and in particular, we spent a lot of time talking about the levels of biological organization, starting with the most simple atoms like carbon and hydrogen and oxygen. And then from there, everything kind of fits together like building blocks, and you start making things that become increasingly more and more and more complex. Atoms come together to form molecules, molecules come together to form cells, cells come together to form tissues, tissues go on to form organs, and then finally, organs and organ systems make up the whole organism. So what we're going to be doing for these next few chapters is examining the physiological importance of some of these tiers of the levels of biological organization. This chapter in particular, we are going to focus on the molecular and atomic basis for human life. So you can look at the topics checklist here. This first video is going to focus in particular on water and hydrogen bonding, and then future videos will focus on important concepts that we're going to need as we trudge ahead into our studies of particular organ systems in the human body. All right, now first, there are going to be things that I don't specifically cover in this set of videos. These are what I call on-your-own topics or OYO topics. I do have old videos in my YouTube feed where I go over these topics if you really want to spend and invest some time listening to me talk about them. But the way I look at it is these are topics that I think you should have seen plenty of times in previous classes, whether it's general biology or general chemistry. I think they're simple enough topics that you can kind of catch yourselves up on them on your own, which is why they're called on your own topics. But of course, if you find you're having trouble with these, definitely try to go watch those other videos that I've recorded in previous years, or just get into contact with me and I'll be happy to kind of get you squared away on some of these topics. Now, some of these topics are going to include things that hopefully you're already fairly familiar with, if not completely familiar with, atoms and subatomic particles. So for a given element, you want to be able to know how many protons does it have, how many neutrons does it have, how many valence electrons does it have. In particular, the number of valence electrons an atom has is going to determine its chemical reactivity. For example, sodium and chloride are able to react with one another to form an ionic bond and form table salt because sodium has 1 valence electron and chlorine has 7. So you definitely want to be able to count valence electrons because that will give you an important clue as to which atoms like to react with other atoms. Ionic bonding I just mentioned, so what kind of atoms like to participate in ionic bonds. We just talked about that with sodium and chlorine. Covalent bonds, very important. Especially the distinction between nonpolar and polar covalent bonds. We're going to talk a lot about polarity in this main set of videos, so just kind of make sure you have the basics of how electrons are shared in covalent bonds down. And then particular types of macromolecules, which we're going to mostly focus on proteins in this main set of videos, but I do want you to spend some time looking into the structure and function of carbohydrates, lipids, and nucleic acids. Now, we will talk about each of those three things at various points throughout the semester, but for the sake of time, I think it's going to be most important that we spend a lot of time talking about proteins, which is why I'm choosing to cover that in great detail in the main videos, and then you can kind of look at my old videos to catch yourselves up on the other three. Okay, let's go ahead and get started with this first video then. Polarity. So the whole deal with covalent bonds, and again, on your own, you can kind of catch yourselves up on this, but to just kind of sum it up, in a covalent bond, electrons are being shared between two atoms. So the best example of this, as we're going to find out, is a water molecule. Water is H2O. It's got 2 atoms of hydrogen and 1 atom of oxygen. So each hydrogen forms 1 covalent bond with the oxygen. So water is a molecule that has a total of 2 covalent bonds. So water ends up being a polar molecule because it is made up of polar covalent bonds. Well, what makes a covalent bond polar? Well, a covalent bond is polar because the electrons that are being shared between the two atoms, in this case, hydrogen and oxygen, in both cases, the electrons that are being shared between hydrogen and oxygen are not being shared equally. It turns out that oxygen is taking up a much greater share of those electrons than either of the two hydrogen atoms are. So because the oxygen atom takes up more than, I guess you would call, its fair share of the electrons, it becomes partially negative charged, right? So each electron has a net charge of -1. So if you're the oxygen atom and suddenly you've got a whole bunch of electrons buzzing about you, you're going to kind of take on more of a negative charge character rather than neutral or positive. And there is a term that we use when we talk about atoms that have a great tendency to pull electrons towards themselves. So in cases like oxygen, we say that those kinds of atoms are electronegative. So oxygen is a very electronegative atom. Hydrogen is not nearly as electronegative. So oxygen takes the electrons because it has a greater desire or affinity for those electrons. Now, in the case of nonpolar covalent bonds and dealing with nonpolar molecules, you end up with nonpolar covalent bonds when you have electrons being shared either equally or almost equally. So a good example of that would be carbon and hydrogen. Carbon has a slightly greater electronegativity than does hydrogen, but it's close enough that each atom, carbon and hydrogen, gets about the same amount of electron around themselves. So in those cases, because the electrons are being shared equally, we would say it's a nonpolar covalent bond. Each atom in the arrangement gets about the same share of electrons, so nobody takes on any partial charges here, right? Okay, so if we go back and look at water again, we've already established that because of oxygen's electronegativity, it takes a greater share of the electrons than hydrogen does. Oxygen takes on a partial negative charge because it's got all those electrons buzzing about. Well, what about hydrogen? Well, hydrogen is experiencing a lack of electrons. So if you normally have electrons buzzing around you and now suddenly you don't, you are actually going to take on a partial positive charge. Because don't forget, at the nucleus of every atom are positively charged protons. So each of those hydrogen atoms has its positively charged proton. Each one of them has one of them. But if they don't have electrons to match it, then the positive charge from the protons becomes kind of overrepresented. So in a water molecule, which, as we say, is the best example of a polar molecule, the oxygen is going to take on very slightly partially negative charges because of all the electrons that are being drawn in towards the oxygen, and the hydrogen atoms are going to get less share of electron, and they're going to be partially positive. So if all of that sounded like a whole bunch of gobbledygook, if you remember nothing else, just please remember that water is a polar molecule. That's going to become exquisitely important as the semester wears on. Now, the reason why polarity is important, especially in the case of water, is that polar molecules like water or like ammonia, which is NH3, nitrogen in that case would be your electronegative atom. Polar molecules like water and ammonia have these weakly positive hydrogen atoms, and they are capable of a very special and very biologically important type of chemical bond, and this is called a hydrogen bond. Now, the important thing to note here is that a hydrogen bond is going to be very different than the covalent bonds that we were talking about previously. So in this picture that you're looking at here from your textbook, we have 3 different water molecules. So there's one, and there's one. The covalent bonds are listed here in a solid straight line. Hydrogen to oxygen, oxygen to hydrogen, this hydrogen to that oxygen, this hydrogen to that oxygen. The hydrogen bonds that you see here in dotted lines are not covalent bonds. These are not electrons being shared. Rather, what a hydrogen bond is a very weak electrical attraction between the partial positive hydrogen and the partially negative oxygen, if it's water, nitrogen, if it's ammonia or something else. Basically, a hydrogen bond is just a very weak electrical attraction between the partially positive hydrogen atom and some other partially negative atom, whether it be oxygen or nitrogen. So in the case of these hydrogen bonds, these are what we call intermolecular forces, meaning that these are attractive forces that exist between two or more different molecules. These are different than the intramolecular forces that exist within a single molecule. So these covalent bonds here that hold the water molecules together, those would be examples of intramolecular forces. Hydrogen bonding is intermolecular forces. They are forces that exist between different individual molecules themselves. So if nothing else, if you need to think of hydrogen bonding in some way, you could think of it as kind of a very, very weak ionic bond. Ionic bonds are basically very strong forms of electrical attractions between positively charged cations and negatively charged anions, like sodium and chloride, but in this case, you don't have full electrical charges. You only have partial electrical charges, so that's why this ends up being a lot weaker. Okay, so hydrogen bonding, we've already established, requires that the electronegative atom, again, whether it's oxygen or nitrogen, it needs to be partially negative so that it can attract that partial positive charge. Well, another thing that we said is that this is a very weak kind of bond. And what that usually means when you're talking about a weak chemical bond is that it's not going to be very strong if you start moving those two molecules apart from each other. So really what hydrogen bonding is ultimately going to require is that the two involved parties, the oxygen and the hydrogen in the case of water, we need them to be fairly close together. So hydrogen bonding really does not end up being possible over very long distances. Well, that actually means something very important for us, because that means that a hydrogen bond can be dissolved by one of two different methods. One you're probably most familiar with, even if you weren't really aware of it, is that you can break hydrogen bonds by applying energy or cranking up the temperature. You do this every time you cook and you boil water. Water is maintained as a liquid in large part because all the water molecules are held tightly together by a network of hydrogen bonds. So that prevents the water molecules from vaporizing and moving far apart from one another. But if you apply heat and you get the temperature of the water up to the boiling point of water, which is 100 degrees Celsius or 212 degrees Fahrenheit, you essentially are applying enough energy to break those hydrogen bonds, that network of hydrogen bonds that's holding all those water molecules in the pot together. You're breaking all those hydrogen bonds and allowing them to separate from each other and evaporate into the air. As soon as you get rid of those hydrogen bonds, each individual water molecule can kind of go off and do its own thing. The other way you can break hydrogen bonds that's going to be very biologically important for us is increasing the distance between the involved atoms by physical separation. You'll find out in an on your own exercise, I think it's in chapter 3, when you kind of take a look at DNA replication inside of cells. Strands of DNA are actually held together by hydrogen bonds. Well, to replicate DNA, you need to peel those two strands apart and break those hydrogen bonds. So it just ends up being a matter of physically separating those two strands of DNA away from each other. Okay, so I mentioned that hydrogen bonds are pretty weak, right? So let's talk a little bit about bond energies so we can get a conceptualization of just how weak they are. First, for reference, let's talk about covalent bonds. Covalent bonds are very, very strong. Covalent bonds are so strong and so rigid that typically, if you want to break a covalent bond, especially inside biological systems, inside the cell, inside the human body, you're going to need an enzyme to do it. Without the appropriate enzyme, the covalent bond is pretty stable. It's not really going to break very easily, right? Well, if you compare a single hydrogen bond to a covalent bond or an ionic bond, which we already established is a much stronger form of the hydrogen bond, a single hydrogen bond is pathetically weak, right? It's really, really weak. It is not at all difficult to break a single hydrogen bond. But hydrogen bonds end up being very important, not because of the importance of a single hydrogen bond, but because of the importance of many, many, many hydrogen bonds at the same time. You just saw an example of this in looking at a pot of water. We have to apply a lot of heat energy to the water to break every single hydrogen bond in that pot of water. You're not talking about breaking one hydrogen bond in that case. You're talking about breaking billions and trillions and gazillions of hydrogen bonds. It takes hardly any energy to break one hydrogen bond, but it takes a ton of energy to break a whole bunch of them all at once. So if for a single molecule, whether it's DNA, which I just mentioned, or proteins are held together in part by hydrogen bonds, as we will find out a little bit later. If a single molecule is being held together by multiple hydrogen bonds, those collective energies, which we already said the individual energies of those bonds are not very great, but if you add them all up, a network of hydrogen bonds becomes cumulatively much stronger than an individual covalent bond. So even though individual hydrogen bonds are very weak, collectively, if you can get a whole bunch of them active at the same time, that can give you a lot of strength and stability for important molecules and important structures inside of cells and inside of the human body. So just real quick, here's an illustration of two strands of DNA being held together by hydrogen bonds. You're going to learn more about this in the On Your Own segment of this chapter, where you look at base pairing rules. Guanine always binds with cytosine, thymine always binds with adenine. But you'll notice here that in each base pair, whether it's A to T or G to C, we have a number of hydrogen bonds existing between the two atoms in the nitrogenous bases. So something we mentioned before, hydrogen bonding can only exist when the atoms are in close proximity to each other, so you have to get these strands of DNA close together in order to get them to stick via hydrogen bonding. And if we want to separate them apart, we need only provide the energy necessary to break these hydrogen bonds, whether that is heat energy to break them apart or mechanical energy to peel them apart. It's not really so much a trivial thing to break many hydrogen bonds at the same time. That's going to require discrete and planned out in advance biological processes for things like DNA replication. Okay, so just to kind of catch ourselves up here, we've already said that water definitely represents the best example of hydrogen bonding in action, and it is for this reason that because water can form so many hydrogen bonds at the same time, it's going to make an ideal solvent for a number of different, very important biologically relevant molecules that we are going to discuss not only in this chapter, but in future chapters as well. So that's where we're going to wrap up this discussion and this video for now. Join us for the next video, and we will continue our discussion of the biological importance of water and how water forms the perfect basis for forming solutions and all of our bodily fluids that contain all sorts of biologically important solutes like sodium and potassium and chloride and glucose and all the good stuff that we are going to need to be aware of as we make our way through the semester. All right, we will go ahead and wrap up here. Thank you for your attention, and I will